Today I'm going to talk about, as Heidi said, a lot of the studies involve people with subclinical neck pain. I'm also going to talk about some of the experimental that we can do to change sensory input from the neck, because both things are very important. Because we're trying to understand how what we do works so that we can do it better and we can figure out who to direct it to. Like Dr. Hodges was saying, we really need to understand who needs what and when they need it. But we also, in terms of understanding how it works, if we can change sensory input in a different way, perhaps through something like muscle fatigue, and study those changes, it gives us another way into understanding this complex problem. So again, Heidi mentioned about maladaptive and adaptive plasticity. So adaptive plasticity is when you get a change at synapses that is beneficial for you as an individual, and maladaptive is when it's not so beneficial. The thing is, we can't always predict what's going to happen. Adaptive plasticity makes elite athletes so elite, and you have amazing musicians, and then on the maladaptive side, you could end up people who have chronic pain. And so trying to understand what causes adaptive versus maladaptive is very, very tricky. I've always maintained a little bit of clinical practice throughout my entire research career. And I remember in New Zealand, this one couple came in, they both had developed this overuse injury they were up at, I think it was their father's cottage batch up in northern New Zealand, and they both had this overuse injury from overusing secateurs in the garden. And one of them was better, a couple of treatments, and the other one was clearly starting to develop a chronic pain problem. So trying to understand what is it that is going to cause that is really critical. So, even in New Zealand, even the lobsters are smart. Okay. So what I'm going to try and talk about today is some studies that the yellow ones are sort of really just trying to understand the phenomenon. What is going on when we change these inputs? And I'm going to talk about some of the things that we know that lead to maladaptive plasticity, like the subclinical neck pain, and then some of the things that we can do to help the brain function better. Because of the audience today, I'm talking about chiropractic in one of the studies, but I've also done some really cool studies for looking at exercise and depression and what changes in those situations as well. Because I think it's all a great big puzzle. And we cannot, some people are great at doing maybe a big world-sized jigsaw puzzle, but I'm just trying to fill in the bits of the puzzle that I know, but what's really exciting is when you can make connections with people working on other parts of the puzzle. Okay, so the effect of neck fatigue on the brain's error signal. So this study, all of the studies I'm talking about today are published, and I'm going to do a little bit, not, I hope, excessive detail, but enough to help you understand why it's not so easy to get some of these studies done. So in this is a study where we wanted to look at neck fatigue. So we know that the CNS uses the position of your head and neck to interpret where your upper limb is in space. Joint position sense is the ability of a person to reproduce a joint position once it has been perturbed. And altered neck sensory input impairs the accuracy of your limb movement in space. We know that passively changing head and neck position can decrease the accuracy of your elbow joint position sense. And muscle fatigue can cause proprioceptive deficits around the joint being tested. And I remember when we first were getting this work published, there's a lot of work out there. If you fatigue biceps or triceps, you are going to impair elbow joint position sense. People kind of understood that was fairly intuitive. But the idea that you could fatigue your neck and impair your elbow joint position sense, for some people, they were very intrigued by that. So what we did was, let's just see if we can, oh great, okay. So we have electrodes on the cervical extensor muscles. That is so we have what I call a myoelectric index of fatigue. Fatigue is literally you stop because it's too hard to keep going. But we can also do things with EMG. We can look at something called the root mean square of the EMG signal, which is the amplitude of the signal. We can also look at the mean and median frequency, and those tell us, they're markers to say, yes, this muscle really was fatigued, right? Because one of the tricks with any kind of fatiguing protocol, people get bored. So they stop trying to fatigue themselves, which is kind of makes sense, right? They're protecting themselves. One of my favorite things in a study was a participant, and I'm not always in the lab, but I happen to be there that day, and I'm saying, come on, you got to keep going, you can do this. And after the experiment, she said, I really like the part where you were encouraging me. And I thought, anyway, it was quite funny, to me anyway. My children tell me I'm not encouraging enough because I just expect excellence as a basic starting point. So, all right, only kidding. Okay, so basically they had to hold their forearm in a certain position, and the reason we kept the range fairly narrow, we wanted to be focused more on muscle spindle feedback, because if your arm is stretched or compressed, you're going to be of course you're activating some of all receptors, but in that narrower range it would be primarily muscle spindles that would be contributing to our awareness of where that joint was. It's very important that you then move the arm back, and then with their eyes closed or blindfolded, they have to reposition the limb, and then we average that. So in the fatigue protocol, I know this looks really dodgy, carrying on with Heidi's theme of 50 Shades of Grey, but anyway, this is just, sorry, okay, this is just one fatigue protocol. We actually find it's easier to fatigue people literally just having their head off the end of a chiropractic table, but this was how we were doing it at the time we were doing that paper. And so they had to hold a 70% maximum voluntary contraction, and as you can see, once for 5 seconds in a row, despite how much we encourage them, they can't maintain that, then that is when the experiment stops. And in our situation, we found that that also always coincided with our myoelectric indices of fatigue. Okay, so actually I'm gonna go back for a minute. I wanna explain something important. So when you do these experiments, it's very, very important to have a control time. And in our case, it takes 4 to 5 minutes to fatigue, so we had to have 4 to 5 minute control period. The reason for that is, even though we know how to do proprioception, so to speak, we don't usually sit there with our elbow in this limited range and do that task. So even though we train them on the opposite arm how to do that, there is an aspect of task familiarization where people get better. And I don't know if anybody follows the literature on exercise and brain plasticity, but there's work that even a single session of exercise can enhance brain plasticity. It's now starting to be discredited, not that it doesn't help, but because a lot of people didn't have an appropriate control, and now they're understanding some of the effects were simply the effects of remeasuring. Okay? And the reason I'm doing all this is because I want to help this audience to think more critically when you're reading research about why you need a control, what is its role. So now this is the work that we did. So here is, prior to fatigue, the average error was around 4 degrees of elbow joint, averaging the 3 repositioning. Then we have a 4-minute rest, which mimics the time frame that it takes to fatigue. So they get a lot better, right? I haven't done anything. If that was a chiropractic adjustment in there, I would think, gosh, I'm good, right? But this is the control. This is nothing happening. So it's just saying they're getting better at the task because they've repeated it. They've become more familiar. You know, in this day and age, a lot of people forget to pay attention to their bodies, so they're actually reconnecting maybe. But then here's the interesting thing. We now fatigue them for 4 to 5 minutes. They get much worse. So this is in the face of having got better. Now they're a lot worse. So now we can actually say that there was something about fatiguing the neck that impaired their ability to actually be good at knowing where their elbow was in space. Now we know that sensory feedback to the CNS from muscle spindles in the neck is altered by fatigue. There's a lot of work on that, well, just from spindles in general. And that distorted neck spindle fatigue would impact the accuracy of your central nervous system map, or the schema of how our brains map or imagery of the body. And so we're going to impact the relationship, the way the brain is perceiving the neck relative to the arm. Now, this is really interesting because previous work that Heidi did in New Zealand, that I was a small part of many years ago, we showed that people with subclinical neck pain following chiropractic adjustments, they actually improved, but their picture prior looked very much like the picture after fatigue. So it's an interesting model that sort of gives us some way to validate and say, when you change input from the spine, you change the way the brain interprets information from the upper limb. So the take home of this is simply that neck sensory feedback could impair upper limb proprioception, and that could possibly then lead to less accurate movements and or injury. So actually, I should have put my slide back in. I have a slide that many, many years ago that related to what patients were saying that even led down this whole track. So I had patients who were saying, oh, they don't come in anymore with pain. I knew I needed to come back because I'm hitting my head getting out of the car again. In my case, I dash around 1,000,000 miles an hour. I start hitting my elbow going through doors, because if you're only allowing yourself a couple of millimeters margin of error, you really notice if your proprioception is less than perfect, right? And here's an interesting one. So Kamal, here's some interesting research we can do. Some people notice that they start biting the tongue or the side of their mouth. So, and these are observations that patients made to me, and it made me start thinking about this whole concept of the body schema. So then the next place, of course, because I like a challenge, is there's not been that much work done with the cerebellum because it's been hard to study. You can now do Fmri of the cerebellum, but it is challenging, and it was an area that has had less study, although we, of course, have a pretty decent idea of what it does from animal research and some human research. So we know that the cerebellum is very important for body schema, for proprioception. It's critical for motor skill learning and coordination, and it can affect the velocity and precision of movements and spatial representations. So the next study that I did was looking at people with subclinical neck pain and looking at a mental rotation study. So again, the body schema is your internal map of your body that is in the brain, and you create that based on past sensory experiences. That is why so a good example, which again is harder in this digital age because we just print a fresh copy every time we want. Anybody ever used like an old-fashioned photocopier? Okay, what happens if you photocopy something 20 times? What does the 20th copy look like compared to the first one? It's not so good, right? It's faded, it's less accurate. This is what happens to your body map if you are not periodically recalibrating it by giving it accurate information from the periphery. Does that make sense? Yeah. Okay. So this internal representation is really critical to guide movement and to interpret the next bit of incoming sensory input. So even small changes to that model could potentially alter your motor skill and your limb mechanics. We know the cerebellum is very metabolic. It's got 50%, more than 50% of the neurons in your brain, important for balance and coordination, but critically it receives and integrates the sensory input that's critical for motor learning, adaptation, forming internal models, and you need it to refine your body movements. So can somebody start the video? Okay, the story behind this video is I had a grad student, her dad's a chiropractor, but she wanted to find a metaphor of what it's like to have disordered sensory motor integration. So this was her metaphor. So it's the idea, it's like literally, it's not quite as extreme as foosball. Is this a Norwegian sport, Heidi? Makes sense, right? Okay. Oh yeah, we don't have the Norwegian commentators there, but okay, one more fall. Okay, that's great, thank you. Okay, so it's just that, it's also would probably be a great way to create some injuries, but it's this idea that if you have this distorted input, how can you possibly make sense of your surroundings and accurately, kick a soccer ball and avoid the people around you? So this paper was looking at what happens when in people with subclinical neck pain and we get them to do a mental rotation task. So mental rotation, for what you don't know, it's not something really fancy, it's not like levitating or anything, it's just your ability to rotate mental representations of 2 or 3D objects accurately. It's important, Glenn, for backing trailers, right? Yes, okay. Yeah, that's an important skill to have if you live in New Zealand, you have to know how to back a trailer. I was not good at that, but that's not why I left the country. Okay. So mental rotation ability is really important for spatially complex skills, object recognition, navigation, problem solving, and action planning. And decreasing, this paper really intrigued me. So there's a kind of TMS, not the kind I do, but you can actually transiently change or decrease activity of parts of the brain with something called RTMS, or repetitive TMS. And they showed that decreasing left cerebellar hemisphere excitability led to slower mental rotation reaction times in healthy participants. So to me that said, hey, the cerebellum is really important in this skill. So what I started thinking about is, could the altered body schema seen in subclinical neck pain extend to altered spatial awareness of objects? So what we hypothesized is that people in the neck pain group would have worse performance on a mental rotation task compared to controls, and this was not just going to be explained by simple changes in movement time. I have a slide coming up, I'll explain a little bit more about reaction time and movement time and how they contribute. And that both groups would improve over time as they got more familiar with the task, but that the subclinical neck pain group would still show slower mental rotation even after a few weeks. These are good. This is what I've learned from my epidemiology colleagues. I used to just do the study, and they said, but how do you even know it's broken? So before you do an intervention study, whether it's chiropractic care, whether it's exercise, you need to show that that population is actually different to start with, and then it makes a lot more sense to do your intervention. So in this case, we had 13 volunteers with neck pain on the chronic pain grading scale and 12 healthy volunteers. This is such a simple experiment. We picked a 2D, not a 3D, because I do hate to admit this, but women are worse at usually, not every single female, obviously, but as a population, worse at 3D mental rotation. So a 2D rotation task, they're equal. And so basically what's happening is, here's a letter R, normal and upright. That's how we measured their simple response time. Basically, the letters B and N are right next to each other on the keyboard, B for backwards, N for normal, and they just had to respond. Is this letter backwards or normal? And you can imagine that as you, oops, wrong one, as you get to the more rotated letters, to the time it takes, you're kind of rotating it. You almost have to rotate it back into congruence before you can tell if it's backwards or normal. And we just measure these response times. So what we found is that this is the baseline for the neck pain and the 4 weeks, baseline for healthy and 4 weeks. So if we're comparing this, the neck pain at 0 and the healthy group at time 0, the subclinical neck pain group was slower at mental rotation. And when we measure them after 4 weeks, they're a bit better, but they're still much slower compared to the healthy group. So that's really important, because now if we do an intervention study in the future, we know there is going to be some improvement just due to familiarity, and so we have to make sure we power that clinical study appropriately to make sure we have enough participants to account for that change just due to them becoming more familiar. But we also know that if you do nothing, they're not just going to get better, because look at this, after 4 weeks, they're still much slower than the healthy participants. So this is just to give you a sense, when you're looking at response times, it actually includes both the recognition phase and the cognitive part of mentally rotating the object, and then the movement phase. That's why it was so important to have just the simple response to the letter R upright. It means they're not moving slower. So even though we tested them on days when they weren't in pain, whenever I present this to my biomechanist colleagues, they always say, but aren't they moving slower because of pain? And then I say, yes, but they weren't in pain. But that being said, it's very critical to have some way to say it's not their movement that was altered in this particular case, it was a more central processing challenge. Okay, so my take home from that was simply that mental rotation ability, which is partly encoded in the cerebellum, along with body schema, is compromised in subclinical neck pain. So that means it could actually change the way we perceive the world around us, even impacting our ability to navigate safely in the world. Okay? All I used to do was just rip my pockets a lot when I went through the door too quickly, hit my elbows, banged into things, but okay. So because I became so interested in the cerebellum, I needed to design some tasks that were good at measuring cerebellar function. So if we look at this particular task, this was a tracking task, and literally this moves down the screen, and what we did was we varied both the amplitude of the waveform and also its frequency, so it wasn't just the same thing all the time, so people had to continuously adapt. And we made the easiest waveforms so that everybody could do them, and we made the hardest ones so they were really challenging, so we had a tool that could allow us to continuously measure improvement. So before we even did any of the research that we wanted to do with that, we actually had to first validate that technique, and then I'm going to describe the next part, which is what we then did with it in our subclinical neck pain group. So that's just showing you what it looks like in terms of the tracking task as the waveform came down, and we measured accuracy, and then we're going to look at the effect of subclinical neck pain. So again, these people on the days they had pain, because remember they had pain-free days, they were between 0 and 2. People who have a 3 pretty much do have pain all the time in terms of the chronic pain grading scale. Again, 24 participants that we divided into 2 groups of 12, And then another important thing, when you're doing motor learning tasks, you need to have a retention period where you measure 24 hours is best, but 24 to 48 hours. Have they retained that skill? Because that's how you measure learning. What you do on day one is just acquiring or acquisition. And in the motor learning world, it's only learning if you show that you can still do it and do it better, and that you can even transfer it to another skill. I'm not going to go into a lot of detail about somatosensory evoked potentials because Dr. Holt will speak more, but very briefly, what we do when we do somatosensory evoked potentials, we stimulate peripheral nerves. In this study, I'm going to be talking about it's the median nerve, but you could stimulate the ulnar, any place you have access to a peripheral nerve from the surface. And what happens when you stimulate, we average hundreds or even thousands of sweeps, and we get these nice peaks at different parts of the brain. And so this N9 is recorded over the brachial plexus. It's very important that that's stable because that is our peripheral volley. That's saying it's not because their arm is in a different position. That's a stable volley. So we know then if we're seeing changes in subsequent peaks, that is due to changes in central processing. This timing, like the N30 here that we're looking at, that means that it's 30 milliseconds after the stimulation. So these are very early peaks. They're what I call precognitive. They're that very earliest phase of somatosensory processing and integration. So this is an example of sort of a pre-post change where somebody's N30 amplitude has been changed. Okay, so in this study, comparing healthy and neck pain people, and we looked at the percent change when they did this complex pursuit task and trained for 15 minutes, and what we found, good, our N9 was nice and stable, that's important. Generally speaking, the peaks all increased, but where I really focused my attention is where were the differences between the subclinical neck pain group and the healthy group, and they were in the N18 peak and the N24 peak. Okay, so the question is, what does that mean? Oh, before I do that, motor performance. So this is also interesting, is that when we did the same day training, the post, both groups improved. We also looked at it, made sure we normalized so that they started at the same point, because as you can see, interestingly, the two groups were, the neck pain group was kind of worse to start with, but they both learned on that day, or at least acquired the skill. But then when we looked at it at retention, what you see, and this is 24 hours later, the healthy group continued to improve and get better at this task, and the neck pain group were just plateaued. They were stuck. Okay? So we know that cerebellar activity increases in the early stages of learning. So the smaller N18 in the healthy group may reflect a decrease in cerebellar inhibition to the motor cortex, because to learn a new skill, you have to transiently release inhibition so that you can encode a new pattern. But then when we get to the N24, which is a different peak, it's the activity from cerebellum to primary somatosensory cortex, the decrease associated in the later stages of learning, and greater ability to rely on well-formed internal body schema. And I was actually thinking of something that Kamal said yesterday, is that the healthy people can rely on their internal model because it's accurate, but the neck pain people less so. And it's a bit like you're saying they could trust their muscles to activate them. So it's quite interesting. So the larger N24 in the subclinical neck pain group suggests that later stage of consolidated learning was not as robust, and a reliance on cerebellar input was still needed. So it's actually harder to do the very same task. So neck dysfunction changes the way the brain responds when learning new movement skills and may even decrease our ability to learn. Okay, so we're on to the last study, and this one does have a chiropractic intervention. So you see how much work it takes to get to the point where you can even do an intervention, because there's an awful lot of things you have to validate and document first. So adaptive plasticity helps the brain learn to function better. So this was just only published in March. And this is, again, looking at a group with subclinical neck pain and its treatment and how the response to motor training and cerebellar plasticity. So before we could even do this, we developed a technique. I won't go into all the details, just enough to give you a sense of what it's about. So if you look at the coil here, let's get this back. Okay, so this is the coil over the motor cortex, activating those corticospinal neurons, and then you get a pathway going down to the muscle, and you can record something called a motor evoked potential. Now, this coil here is over the cerebellum. The cerebellum doesn't have a direct output, but if I stimulate the cerebellum 5 milliseconds in advance of this one, I can actually partially inhibit that motor evoked potential. So what we did is we came up with a technique to individualize what 50% inhibition was for each person, and then we could have a tool to compare differences in cerebellar inhibition between people. So we hypothesized that people who had subclinical neck pain would show more inhibited at their 50% level after they did a motor acquisition task than healthy participants due to this altered neck input, and that a single treatment session would normalize sensory input and restore this cerebellar response. So again, we had 27 people with subclinical neck pain, and we divided them into two groups. We had healthy volunteers, so we knew what healthy looked like, and we randomized the subclinical neck pain to either manipulation or passive head movement. So again, this is the stimulation from the two coils. The one thing I did want to show you here, so if you have the red dots would be a normal motor evoked potential. When you stimulate the cerebellum first, you should see it inhibits by roughly 50%. So the two conditions for adjustment versus control, the adjustment involved diversified technique, and this particular study, because we had to literally have them, okay, you like this study too, this story, it was a lazy boy chair that had to be reclined, so that's why we stuck with neck manipulation. And when I first arrived in Canada 10 years ago, I don't have this chair anymore, sadly, but I was setting up my lab, I'd spent all my research startup funds, and I said to my mother, I said, I need a secondhand lazy boy for my experimental chair. So I just think nothing of it. Okay, the next day, I'm at the church garage sale and the priest's chair is for sale. So anyway, the priest's lazy boy was my experimental lab chair for 8 years. And she made some poor old guy carry it from the church thing and put it in her truck. It was just quite funny. Anyway, so passive head movement, this was not meant to be a sham in any way. It was meant to control for the physiology that we're touching, we're moving the spine, just the fact that time is passing, all of those things, very important to have a control for that. And the touch, right? The motor task in this case was, because this was actually done before the other study, we just had them type, they had randomized sequences of letters, Z, D, P, F, that they had to type on a keyboard using their first dorsal interosseous muscle here. Okay. Oops. And the reason we picked this task, same as the tracing task, similar tasks have been shown to activate the cerebellum using both MRI and PET scans. Okay, so here's some of our raw traces, and I'm going to get to the group results, but the main thing I want to show here is our pre. There in our healthy group there's the top, the bottom, our peak to peak of our motor evoked potential. This is our inhibition, which we want to see this inhibition. After you learn a new skill, you transiently see this total loss of inhibition because you have to disinhibit to learn. Now the neck pain control group, they showed no disinhibition. And then look at the group that got the treatment. They actually showed this massive disinhibition. So if we look at that in terms of the group results, what we actually see here, this 100% line is your normal motor evoked potential amplitude. The red is your inhibited motor evoked potentials. And then the green line is after you do the motor learning, you disinhibit, and it almost looks like there's been no cerebellar stimulation at all because the cerebellum, those pathways to the motor cortex are disinhibited. This actually shocked me, this next slide. I thought I would see less ability to disinhibit, okay? But instead, what I found was no disinhibition. The brain of people with neck pain in the absence of treatment did not disinhibit at all when in response to learning or acquiring a new motor skill. And then this is the group after the neck manipulation, spinal adjustments. There's your 100% line. There's your normal cerebellar inhibition. So not only was there disinhibition, it looks like there was facilitation. I don't think there was true I think the facilitation was the central nervous system trying to cope with the inability to disinhibit, and that we're seeing kind of a combined, and I suspect that you wouldn't see that after they had once the capacity to disinhibit had restored. Another problem when you're doing motor learning experiments, when you learn a new skill, the cerebellum only disinhibits the first time you learn it. Then it starts to encode and store in other places. So you have to continuously be developing new tasks if you want to continuously be studying changes in cerebellar plasticity. So it's actually quite challenging area to work in. So, neck pain, no cerebellar disinhibition, slower response times, which I didn't even show you those results, versus healthy. A single treatment session increased the magnitude of cerebellar responses while improving response time, or at least the magnitude of cerebellar disinhibition. Now, we measured this about 20 minutes after treatment. Future work needs to look at how long this cerebellar response is influenced for. And so subclinical neck pain decreases the ability of the cerebellum to respond appropriately when learning new skills. And chiropractic treatment restored the capacity of the cerebellum to respond appropriately. So just to sort of fill in the model, so this is kind of the scientist version of the model that Heidi presents, which is also very scientific but uses chiropractic language. But I get most of my funding from the Natural Science and Research Council of Canada because I'm studying mechanisms. So this is the model that I work on, is the idea that altered sensory input from your neck, whether it's fatigue, postural stress, joint dysfunction, including subluxations, that can alter your body schema, which leads to altered processing of upper limb sensory information, alters your sensory motor integration, and then your motor control. Now, altered sensory motor integration may also impair your ability to filter sensory input generated by repetitive activity. It could be the link between, say, altered neck inputs and people who get overuse injuries. And then the altered motor control is going to impair your ability, and so will this, to learn and perform motor skills. Now, this is the side that I haven't really looked at, although it's why I was interested many, many years ago in practice, is that may then lead to all of these different chronic pain things, or at least be contributing to them. So, thank you for your attention. And I would just like to thank, again, all my collaborators and the wonderful graduate students I have. So, thank you.